Okay, so this is chapter 12. We're going to talk about the cell cycle. And so chapters 12 and 13 kind of go together. One talks about mitosis, one talks about meiosis. So in class, we'll be talking about these two chapters together. I will split them up into two separate lectures and two separate videos here, but you should probably watch them together and think about them together in terms of preparing for class. All right, so a little bit of an overview of this chapter. What we're going to be talking about is the fact that DNA is the hereditary information, or contains the hereditary information, which in our case we're going to be talking about in terms of chromosomes in this chapter. We know that before cells reproduce and divide, before they reproduce, they have to undergo division. So we're going to be talking about two different types of division. So in this chapter, in chapter 12, we're going to be talking about mitosis, and in chapter 13, we will be focusing on meiosis. So we know that multi-celled organisms can undergo growth and repair themselves through the process of mitosis, and so that's what we're going to be focusing on here in this chapter. So mitosis and meiosis are both nuclear division mechanisms, and so this is something that is really important, that we understand mitosis and meiosis are nuclear division. So a lot of times people use this term to mean cell division, and you're going to see that it's not actually cell division. So as we go through the processes, I want you to make a distinction between nuclear division and cell division. There are distinctions, and there is a separate process that we call cytokinesis that actually constitutes cell division. So that's something I want you to pay attention to. When we talk about mitosis, we talk about mitosis happening in somatic cells, which are basically the body cells. Any cell within your body, your kidney cell, a cardiac cell, a muscle cell, a liver cell, all of those are what we call somatic cells. So mitosis occurs in somatic cells, and that allows for things like growth, repair, and even asexual reproduction. In chapter 13, when we talk about meiosis, that is going to occur in what we call the germ cells. For humans, the germ cells are the gametes, the sperm and the egg. So that is where meiosis occurs, so producing these gametes, and allows for sexual reproduction. So we will talk a lot about distinctions between mitosis in chapter 12 and meiosis in chapter 13. We know that prokaryotic cells only reproduce asexually, so these are going to be cells that undergo mitosis. When we talk about meiosis, we're talking about the eukaryotic cells. All right, so a couple of reasons why cells would need to divide. Why would they need to undergo mitosis or meiosis? Well, the first reason is to increase body size. So we would talk about this in terms of growth. So certainly you have more cells now than you did when you were an infant. And that is because as your body has grown, your cells have had to divide and multiply so that you have more cells to increase your body size. The second reason why cells would need to undergo division is to replace worn out or dead cells. So, for example, your red blood cells, which are responsible for carrying hemoglobin to every part of your body, have a lifespan of about 1 month. Okay, so these red blood cells, after about a month of doing this job, they're worn out. They need to be replaced. And so your red blood cells are constantly dividing and replicating in order to replace out those worn out cells or dying cells. The third reason why cells would undergo division is to repair damaged tissue. So one interesting area that's getting a lot of research in this recently are spinal cord injuries, as well as traumatic brain injuries, or TBIs. So certainly as servicemen and women are coming back with traumatic brain injuries and potentially even spinal cord injuries. There's a lot of research looking into how do we increase the rates of cell division in circumstances like this where there has been damage so that we can repair that damaged tissue. And then again, probably the last and maybe the most obvious reason why cells divide is in order to pass on the genetic information to their offspring. So in this case, we are really focusing on meiosis. So we have production of the egg and the sperm that are produced through the process of meiosis so that fertilization can occur and information can be passed down from one generation to the next. So again, we will talk much more about meiosis when we get to the chapter 13 lecture. Okay, so before we get into the process of nuclear and cell division, we need to have a little bit of terminology under our belt and a little bit of vocabulary. And so one of the key things that you understand as you go through what happens in the process of mitosis and meiosis is that you understand what is happening to the chromosome structure. So there are two types of chromosome structures that we need to recognize. The first one is unduplicated and the second one is duplicated. So this first picture right here that I'm going to label number 1, this is one chromosome that is in an unduplicated form. So in a minute you're going to see when chromosomes undergo duplication during what we call prophase. So this first picture is prior to prophase. The cells have not undergone, the chromosomes have not undergone duplication yet. This second picture that I'm labeling number 2, you see, is also still one chromosome. It is now just in its duplicated form. Where a lot of students mess up is they call this 2 chromosomes, and that is not the case. It is still one chromosome. It's just in a duplicated form. Now, even for this duplicated chromosome, we have a couple of terms that we use here. So for each of the arms of this chromosome, they are referred to as a chromatid. And that TID is important at the end. So one is called the chromatid, the other is called the sister chromatid. Okay, so the whole thing itself is a chromosome. Okay, that refers to the DNA itself. And then we have one arm that is a chromatid, the other arm is the sister chromatid. So chromosome, by the way, is singular. So when we talk about one chromosome, we would use that term. When we talk about all of the DNA inside the cell, sort of the plural form, we use this word, chromatin. Okay, and that's why that TID is so important at the end there. Because when we talk about chromatin, that is the plural form. So that would mean all the DNA inside a cell. So you have to keep those terms separate and distinct. So here's a picture of a chromosome, a photomicrograph of one chromosome inside the cell. So you can see highlighted in pink, this would be one chromatid, and this would be the other chromatid, or the sister chromatid. And you can kind of see there's this area in the center where it looks like you've taken and pinched them right in the center. We call that the centromere, and you're going to see why that's important in just one second. But that pinched in center, or that pinched in part right in the center, we call the centromere. So we need to keep all of these terms separate as we go through the rest of the processes. Alright, so let's talk about chromosome number, for example. So we have a couple of terms that we use to define a cell based on the number of chromosomes. So if we say that a cell is diploid, and by the way, when we are talking about these specific terms, we always talk about them in terms of cells. So a cell is diploid, never a chromosome or a nuclei or anything else, when we use this term diploid, we are referring to the entire cell. So that means if we say that a cell is diploid, it means it has 2 of each type of chromosomes. So in humans, you have 23 pairs or 46 total chromosomes. You have 2 of each type. You got 1 from your mom and you got 1 from your dad. And so you can see here we put those chromosomes in pairs. We group them in pairs. And so that tells us that this is a diploid cell. So we have chromosome number 1, chromosome number 2, and chromosome number 3, chromosome number 4, chromosome number 5, and so forth. And you see we have 2 of each type. This occurs in all of your somatic cells. So all of your somatic cells are what we call diploid. The only cells in the body that are not diploid are the sperm and the egg cells, or the gametes. The gametes are what we call haploid. And haploid, so again, these are our gametes, that means that they have 1 of each type. So we would have 1 chromosome 1 and 1 chromosome 2 and 1 chromosome 3 and 1 chromosome 4 and 1 chromosome 5 and so forth. So the gametes have 1 of each type. So in humans that means we have only 23 chromosomes in a gamete cell, and that occurs in the sperm and the egg cells only. Okay, every other cell in your body is diploid. Okay, so let's talk about the phases of the cell cycle. There are 3 sort of main phases that we talk about. The first one is what we call interphase. Then we have nuclear division, so that's mitosis or meiosis. And then lastly, we're going to have cytokinesis, and that is what refers to the cytoplasmic division. So that is why we make a distinction between cellular division and nuclear division, because we have this third step of cytokinesis, and we'll walk through what that looks like in just a minute. Okay, so let's first of all talk about interphase. So a cell spends almost all of its time in interphase. You can see what I'm highlighting in blue here sort of represents interphase for the cell cycle, and you can see that about 75 to 80% of this circle is interphase. And so a cell spends most of its time in interphase, and there are 3 subphases of interphase that are characterized by cell growth, DNA duplication, and preparation for division. So the first part of interphase we call G1. This is sort of the first subphase of interphase. This is a period of cell growth. During G1, the cell itself expands. There's more room created in the cytoplasm. There is just a lot of growth within that cell. So the DNA hasn't duplicated yet, but this is characterized primarily by a period of growth. So a cell would go from something like that to something like that. The second subphase of interphase is called the S phase. So this you could think of as standing for synthesis, because this is where the DNA is duplicated. All right, so we synthesize a whole new copy of all the DNA inside that cell. So that means that the chromosomes get duplicated. So remember, in G1, those chromosomes look like this. They are unduplicated. And by the end of S phase, our chromosomes are going to look like this, where they are duplicated, where we have our 2 chromatids. The third phase of interphase is called G2, and this is really just a period after the DNA is duplicated. Again, the cell is preparing for division, so all of the organelles are being copied, and we are preparing everything that cell is going to need to divide into 2 fully distinct independent cells. All right, so from interphase, we enter either mitosis or meiosis. So again, in this chapter, we are focusing on mitosis. And so we have just finished G2 of interphase, and we are entering mitosis. Mitosis itself has 4 subphases, the first of which is called prophase. So there are a couple of things that happen during prophase. I'm going to walk you through these, and then I'll show you a picture of what it looks like. So the first thing that sort of happens is the chromosomes become visible. Up until this point, the chromosomes are very stretched out, very thin, very, very narrow, and difficult to see under a microscope. But when prophase begins to happen, those chromosomes condense and shorten and thicken up a little bit and become very visible under a microscope. So we can see we can actually visualize the chromosomes during prophase. The second thing that happens is we form what's called a spindle apparatus. This is what's going to actually do the work when it comes time to separate the chromosomes into 2 distinct nuclei. And I'm going to show you a picture of that in just a second and what that looks like. The third thing that happens is that the nuclear envelope begins to break apart. So if you remember back from chapter 6, we talked about the fact that there is a membrane surrounding that nucleus. Well, in order for cell division to occur, in order for nuclear division to occur, we have to break apart that nuclear membrane. And so that begins to occur during prophase. Okay, so here's a picture of what's going on here during prophase. So here we are going from early prophase into late prophase. So you can see how the chromosomes have changed. So if you look here, they're very thin, they're very wispy. And if we look by late prophase, those chromosomes have condensed, they've shortened, they've thickened. And if you notice, these chromosomes are in that duplicated form where we can visualize that centromere, because they have already undergone interphase, which is where chromosome duplication occurs. So the chromosomes have become visible. The second thing that happens is the spindle apparatus begins to form. So we have these 2 little protein subunits here that are called centrioles. So these are called centrioles. And basically one is going to be at one end of the cell and the other one is going to be at the other end of the cell. So this guy is working his way down to the opposite pole of the cell. And out from these centrioles, we have these long fibers called microtubules. So microtubules are long protein fibers that are going to actually do the job of pulling apart the chromosomes when it comes time to separate them. And then the last thing that happens during prophase is that nuclear envelope starts to deteriorate. So you can see here we've got a few little holes in that envelope, but by late prophase, that nuclear envelope has become very spotty, and there are gaping holes, and it's going to continue to deteriorate until it is gone. All right, the second subphase of mitosis is called metaphase. Metaphase is probably the easiest one to recognize under the microscope. So when you're looking at cells in lab under the microscope and trying to identify phases, metaphase is probably the easiest one to see. So a couple of things happen here. The chromosomes begin to interact with those microtubules on the spindle apparatus, and we'll talk about that. The microtubules are going to attach at those centromeres, at those sort of pinched in parts of the chromosome. The microtubules are going to start to pull on the chromosomes and orient those chromosomes along the midline of the cell. So the alignment of chromosomes happens right along the midline of the cell, and we orient those sister chromatids to opposite poles. So I'm going to show you what that looks like in the next slide. Okay, so here we just finished up prophase over here on this side, and we are transitioning over here to metaphase. So again, here's our centrioles. This guy's still working his way down to the bottom of that cell. So you can see here it has reached there. So we have these centrioles. We have microtubules that are attached to each of these centrioles. So, and they send out these microtubules, so they attach right at the centromeres of the chromosomes. So you can see we have right at that pinched in part where the microtubules are attached. They attach right to the centrioles. And the other thing that happens is we get these chromosomes lined up right along the midline. So again, that's one reason why this is so easy to visualize under the microscope, because you can very clearly see the chromosomes will be lined up right along the midline of the cell. You can also see that we've oriented these chromosomes so that you can kind of almost go ahead and visualize that one sister chromatid is going to go this way and the other sister chromatid is going to go this way. So we can see what we're actually going to do here is, we'll see this in the next couple of slides, we are going to separate the sister chromatids. Okay, so keep that in mind. That is so important that we are separating the sister chromatids. Okay, so here we are in the third subphase of mitosis, and this is anaphase. So anaphase is also an easy one to recognize under the microscope, and that's because we start to see, or we do see, movement of those sister chromatids to opposite poles of the cell. So here we still have our centrioles, we have our microtubules, but you notice that the microtubules are much shorter in this picture than they were in the previous picture showing you metaphase. And that's because that's the way that we separate those sister chromatids, is those microtubules start to shorten, and as they shorten, they pull apart our sister chromatids. So one sister chromatid goes to one end of the cell, the other sister chromatid goes to the other end of the cell. So this is just showing you the action of those microtubules. So we have our centrioles here, and then out of those centrioles we have microtubules that attach at the centromeres of each of those chromosomes. So here the microtubules are very long and stretched out. What happens is those microtubules shorten, and as they do so, they pull the chromosomes toward the poles so that they stay attached at that centromere location, and as the microtubules shorten, they separate the sister chromatids, pulling them to opposite ends of the cell. All right, the last subphase of mitosis is telophase. So again, this is still mitosis. And telophase is where the chromosomes arrive at the spindle poles, so up here, back up where the centrioles were. The chromosomes make their way back up there. They return to that sort of thread-like form where they're very stretched out, very spindly, very hard to see, and the new nuclear membranes form. So by the end of telophase, we have completed nuclear division. So nuclear division is complete. Not cellular division, but nuclear division is complete. So here we are in telophase. We have one cell right now with two nuclei. Okay, so here's our one big cell. This is one big plasma membrane, and we have two newly formed nuclei. Now this cell cannot stay like this. We have to move on to the third phase of the cell cycle, which is cytokinesis. All right, so let's talk a little bit about cytokinesis then. So cytokinesis is what actually involves division of the cytoplasm. So we have separated our nuclei. We have two newly formed nuclei, and now we have to separate the cytoplasm so that we end up with two fully new functional independent cells. This happens a little bit differently in plant cells versus animal cells. So in plant cells, we form what is called a cell plate. So it is a very thick wall of vesicles that form right here along the spindle. Oops. Right here along the equator of the cell. So we form what's called the spindle equator. So we got all these vesicles that converge here, and they sort of combine and get closer and closer together until they form what's called the cell plate. And that cell plate reaches all the way across that cell until we get two new cells. So you can kind of see this here in this picture. You can kind of see that cell plate forming here, and then in this picture you kind of see that we have distinctly two new cells formed here. Now, in animal cells, it happens a little bit differently, where we get the formation of what's called a cleavage furrow. So this is what happens in animal cells. This is distinct from the cell plate formation in plant cells. So basically what happens here is you have a microfilaments that come in and just beneath the plasma membrane, and they contract. The microfilaments sort of push in on that plasma membrane, so until we get something like that, where it just cuts that thing in half, and we end up with two new cells. So you can sort of think about this as taking a piece of Play-Doh and just putting pressure on it in the middle of the Play-Doh until it breaks apart into two pieces. And that's exactly sort of what happens here. These microfilaments are just putting pressure on that plasma membrane until it completely divides and cuts that cell in half. Alright, so there are lots of good videos out there on mitosis, so I encourage you to watch those. This is just one example, but there are many, many good videos. If you go to YouTube and Google mitosis, there are lots of great videos that would show you this process in general, and we'll watch a couple in class when we talk about chapter 12. The one thing that I want you to remember about mitosis when it comes to chromosome number, there are a couple of things that are important. So mitosis does what we call maintains chromosome number. That means if our parent cell has 4 chromosomes, our daughter cells are going to have 4 chromosomes. If our parent cells have 46 chromosomes, our daughter cells are going to have 46 chromosomes. In mitosis, you also get what are called genetic clones. The daughter cells are genetically identical to the parent cells. There is no variation, there is no distinction. The DNA that gets passed down in mitosis from parent cell to daughter cell is exactly the same. They are genetic clones of the parents. So those two things are really important to remember about mitosis, because they are distinct from meiosis, which we will talk about in chapter 13.